and welcome back to the second part of muscle anatomy. In the first part, we looked at the structure, particularly of skeletal muscle cells. That's important because we need to understand the unit within the muscle cell, the sarcomere, and how the actin and myosin are arranged, because these are the two proteins which slide past each other in the sliding filament theory of muscle contraction. And we'll come back in a later topic to actually look at that in more detail. Right now, because we have an understanding of the structure of skeletal muscle, we can now consider how that structure relates to neural control. This whole topic is neuromuscular anatomy, so we're going to focus on how the nervous system actually attaches onto these skeletal muscle cells and what information is being delivered to the muscle and what information is being removed from the muscle. So an important concept that we're going to come across here is proprioception. I'll talk about that in more detail in a few minutes. So, neural control of skeletal muscles. So, within the nervous system, you have information flowing into the central processing area, which is the central nervous system, the spine, and the brain. That is considered the sensory side, and then the neurons within the spinal cord, and particularly the cortex of the brain itself, are able to take that information and make decisions based upon it. And then it sends back signals through the motor side to the skeletal muscles through the somatic nervous system, specifically the voluntary nervous system that controls our skeletal muscles. And these neurons that are taking that information from the brain, in particular, to the skeletal muscles, is called the motor side, motor neurons. To get a signal from the brain to the muscle takes two neurons. The upper motor neurons carry the information from the very area of the brain the decision was made, all the way down to where the part of the spinal cord that the second neuron is going to leave to actually access the muscle directly. So this upper motor neuron synapses with the lower motor neuron, which carries a signal from the spinal cord out to the muscle and attaches directly onto the muscle. So lower motor neuron activity can be influenced by feedback from the muscles and the tendons themselves. This is actually a very important feedback because this is proprioception. So what is proprioception? It is a sense within the body that allows your brain to know how long every one of your 630 odd muscles are at any given time. This doesn't just let the brain know how contracted the muscle is. It also allows the brain to understand where the body is in three-dimensional space. Regardless of whether we're floating in space, upside down, etc., we still know where our feet are, our hands are, etc., without needing to look. Proprioception is really a large part of our motor learning process when we're picking up a new sport, and muscle memory is associated with that in order to fine-tune the muscles in order to be ready to act in the appropriate way faster as we train. So how is that done? It's sensory feedback from the muscle itself and also from the tendon attached to the muscle. This can have different effects on the upper motor neuron. It can facilitate stronger signal from the motor neuron in the upper regions, or it can inhibit the stimulus depending on need. So let's move on. So there are two structures that are actually involved within a whole muscle, and these are muscle cells which have in this case, the muscle spindles have been modified and are no longer acting as contractile units. Instead, they're like having a tape measure attached inside every one of your muscles. So to control skeletal movements, the brain has to have constant feedback saying exactly how contracted any given muscle is. So the information is sent back from the tendons through the Golgi tendon organ. So this is embedded into the tendon itself and is telling the brain how much tension there is on the tendon. Now this obviously is important because too much tension on a tendon could cause the tendon to break, and that's a serious injury. The muscle spindles are within the muscle itself, and they're like the standard muscle cell, but they have specialist structures inside them instead of the normal myofibrils that we just looked at in the last lecture. So the muscle spindle is measuring the length of the muscle, how contracted the muscle is. So these cells are thin, and we refer to them as intrafusal fibers. The extrafusal fibers are the contractile muscle cells that we associate with muscle. So these intrafusal fibers attach into the tendon at either end of the muscle, so they run the whole length of the muscle. And their contractile apparatus is absent from the middle part of them. Instead, they have fibers which are referred to either as nuclear bag fibers or nuclear chain fibers. Now, our need to know all about the inner structures of nuclear bag and chain fibers are not great at this point, but knowing that they exist and how a little bit on how they actually are operating, it would be helpful. So, sensory neurons. In order for the brain to know anything that's going on in the muscle, it needs to have sensory neurons picking that information up and carrying it back to the brain so the brain can make decisions sent back down the upper and then lower motor neurons, which cause contraction. So we have primary and secondary sensory endings, and again there's more information within the slide, but I really just want you to take the understanding that there are sensory neurons picking up the information from the muscle spindles and the Golgi tendon apparatus. Any sudden rapid stretching of a muscle causes the spindles themselves to be stretched, stimulating both the primary and the secondary sensory endings. This produces a forceful muscle contraction. So let's think about that again. So you're stretching a muscle, you get a sudden rapid stretch of a muscle, but that produces a more forceful muscle contraction. Well, that muscle is being stretched, so now it's shortening. This is a protection mechanism built in to try and prevent damage to the muscle. So a very easy way to give an example of this is when I was young and we were stretching out before playing a sport or something like that. I mean, it was often people would like lean over, like to touch their toes, and kind of like straighten their legs, and they would bounce up and down, like trying to touch the toes and release, and touch the toes and release, and they bounce backwards and forwards on their in the stretch. And this is actually creating the rapid stretch sensory information, which then actually caused their hamstrings, which are being stretched at that time, to shorten, to contract. So instead of stretching muscles out, warming up to get ready to compete in sports, we were actually shortening the muscle and making them more likely to tear because we were bouncing on the stretch. So as I'm sure you're all aware, any of you that are involved in sports, you never bounce on a stretch. You never you hold the stretch, you move out to the stretch, and you hold it in place. You don't release and then go back and release and go back, and it's because of this reason in the muscle spindles, you'll stimulate both the primary and secondary sensory endings, and this will cause a forceful contraction of the muscle that you're trying to lengthen and completely go against what you were trying to achieve, and also sets you up potentially for a serious injury. So those extrafusal fibers, remember, are our ordinary muscle fibers. They are not part of the spindles, they just cause our muscle to shorten. So we have two types of motor neurons actually attaching to our muscles. We have the alpha motor neurons, which are doing the job of stimulating our normal extrafusal muscle fibers to contract. But we also have gamma motor neurons, and these attach on to the intrafusal fibers. They cause an isometric muscle contraction because they're holding the spindles at the same length as the contracting muscle cells around them, and that allows the brain to know exactly what the length is of that muscle at any time. So as the alpha motor neuron is stimulating contraction of the muscle, gamma motor neurons are making sure that the muscle spindle is measuring that contraction. So stimulation by alpha motor neurons is the only way to make a skeletal muscle contract. The gammas are just there as part of that measurement system. And diagrammatically, you can see there's muscle spindle, right there. As we look into it, you see it looks quite different inside with the nuclear chains and the nuclear bag fibers sitting in there, which are being innervated by gamma neurons. A little bit of muscle contraction. It's just enough to kind of keep it up with the shortening of the muscles around it. So, co-activation of alpha and gamma motor neurons, the ones that attach onto the muscles. The alpha is causing the muscles contraction, the gamma is measuring that length of that contraction using the muscle spindles. So upper motor neurons usually stimulate both alpha and gamma motor neurons simultaneously to keep everything together. As I said, stimulation of alpha motor neurons, you get muscle contraction. Stimulation of gamma, you get intrafusal fibers measuring that contraction. So activity of the gamma motor neurons is there to keep muscle spindles under the proper tension while the muscle is relaxed, and so you're still getting a measurement through the sensory endings. So this kind of takes us on to like a few minutes of understanding reflexes and why we use reflexes. So there are different types of reflex that we see. Now I think the universally known reflex is the knee-jerk reflex, and this is also one of the very simplest because it's a monosynaptic stretch reflex. What does that mean? Well, mono, one, synapse, a junction between two neurons. So there's just a sensory neuron going out and a motor neuron coming back, and that's it. It is a very, very simple mechanism. It's very, very fast, and it's there for safety and security, so you don't overstretch a muscle. So if you hit a tendon on the insertion side of a muscle quickly, it causes a sudden stretch on the muscle itself. That signal is sent through the sensory neuron to the spinal cord synapses with an alpha motor neuron, which then is attached to that muscle, which causes it to contract. And so you get the knee jerk as when you do this to the rectus femoris muscle, because that's its action anyway, but you get this shortening of the muscle as a result. A slightly more complex reflex is the Golgi tendon organ. So notice this time it's a disynaptic. There's two synapses there because there's a little bit of an interneuron, which is a processing neuron, and it can actually have the ability to change the outcome. In a monosynaptic, you stretch, you get your contraction of the muscle that was stretched. In a Golgi tendon organ reflex, you can actually get a slightly different result. So you get the Golgi tendon organ being stretched, signal sent in, then you get an inhibition of the alpha motor neuron to allow that stretch to continue. So, and if you think about it, this is where the tendon is being overstretched, and the tendon needs relief from that overstretch. So if you were to use a monosynaptic reflex, you pull even harder on that tendon because the muscle is now shortening. Instead, you use that interneuron to kill the signal, inhibit the signal, and therefore the alpha motor neuron is inhibited, and then the muscle relaxes and allows the muscle to lengthen and that takes the tension off the Golgi tendon organ. It relaxes the tendon as well. So the Golgi tendon organ reflex protects the tendon, whereas the monosynaptic reflex protects the muscle. So, a few more. Reciprocal innervation. So this one looks a little bit more complicated. So you're hitting the insertion point of the muscle near the tendon. You're getting the sensory neuron going in. Now the sensory neuron is synapsing with a monosynaptic reflex to the muscle, which causes it to contract, but at the same time it's synapsing with an interneuron, which causes inhibition of the antagonistic muscle on the other side, which causes it to relax. So if it's an extensor, such as the tricep, it causes the bicep to relax, to not interfere with the shortening of one side. Crossed extensor reflex, my personal favorite, because it saves me a number of times, because yes, I have an ability to stand on things which are sharp without shoes on. So this is obviously, as you can see, a much more complex reflex system, but it's double reciprocal. And the important thing is it's affecting muscles on the contralateral side of the spinal cord as well as the ipsilateral side, which is what we've shown so far. So in anatomy and sports science, etc., ipsilateral is on the same side, contralateral is on the opposite side of the body. So you're walking along, going to the bathroom at night. I mean, this could be a pin here, but it could be in my experience, it's Lego bricks which do most damage. I've stood on far too many in my personal life. Anyway, so you're walking toward the bathroom, lights are dim or off, and your foot comes down onto a Lego brick. That sensory signal is sent in through the spinal cord, and look, it's synapsing in 3 places. So 2 of the places are on the ipsilateral side. The first one stimulates the extensor and flexor. So it stimulates the flexor to get that foot up off the ground. The extensor is relaxed to allow that movement to take place. And then on the opposite side, it stimulates with an interneuron, which does the opposite effect on this side. The extensor on the contralateral side, this foot, in this case the left foot, is contracted and the flexor is relaxed. So you're basically shifting your weight off one side onto the other through this crossed extensor reflex to prevent more damage from taking place. So, upper motor neuron control of skeletal muscles. So the upper motor neurons control the lower motor neurons, and they do this through tracts. Tracts are bundles of nerves running up and down the spinal cord, and we have the pyramidal tracts and the extrapyramidal tracts. So the pyramidal tracts are running from the precentral gyrus, and they are cross over to the contralateral sides of the pyramids of the medulla. Extrapyramidal tracts and the neurons in the other areas of the brain. So these tracts are coming down from the brain, and they're affecting the lower motor neurons and therefore affecting the muscle contraction and movement and coordination of that. So how are we coordinating that? Mainly through the cerebellum, an area of the brain at the back, separated off from the rest of the brain. It's the bump, and you're kind of at the occiput area. The cerebellum takes sensory input from the muscle spindles. It takes input from the Golgi tendon organs and areas of the cerebral cortex devoted to vision and equilibrium. So what's that meaning? So a balance, and this is basically describing balance. Balance in our bodies is achieved through 3 things: our vision, the vestibular apparatus in our ears, and the knowledge of how long each one of our muscles are. So The cerebellum receives information on all of these. The proprioception is sent up through the sensory nervous system, on the somatic nervous system through muscle spindles and Golgi tendon organs, and then we get information from the visual cortex and from the vestibular apparatus in our ears. There are no descending tracks from the cerebellum directly, meaning that the cerebellum is not connected to any of the rest of our body outside of our brain. The cerebellum is kind of like a processing center for the brain. It takes the information in, but then sends it back out to the brain, to the motor cortex for action. So all output from the cerebellum is actually inhibitory, and that helps to aid the motor coordination. So as the information comes in, the cerebellum is essentially deciding which muscles to switch off to allow for certain movements to be carried out successfully. Another part of the brain, the basal nuclei. And inside the basal nuclei, there are many different structures. I should really have had a diagram for this. But the caudate nucleus, the putamen, globus pallidus, and the nuclei of the thalamus, substantia nigra, and the red nucleus are all parts of the basal nuclei. Don't worry, you don't have to learn them. And they have profound inhibitory effects on the activity of the lower motor neurons. Damage to this area results in increased muscle tone. So the upper, without getting into too much detail, particularly as a substantia nigra, this is where Parkinson's develops from. Substantia nigra stimulates the upper motor neurons via the dopamine effect, and so dopamine is a neurotransmitter there, and as that drops down in Parkinson's, there's less and less control over the upper motor neurons, which naturally are overstimulated. So at rest, our body, without the control of the basal nuclei, we would just basically be moving around all the time because our muscles are being told to move around by the brain more than they should be. And then this basal nuclei calms everything down and says, no, you don't need to be doing that, just stay. So you can sit at rest, not moving, which is why someone with Parkinson's at rest has a resting tremor. And it tends to cycle at 5 hertz as a result of that. There are other conditions which are even worse than that, but that's not the scope of this course to get into. Hodgkinson's being one of them. Anyway, so basal nuclei calms down the signals coming from the higher function of the brain to the upper motor neurons to make basically the muscle movements coordinated and smooth, as opposed to jerky and overcompensated. And that's the end of this slam, so thank you for listening.